My name is Asha Korecka, and I'm very grateful and honored to be here today. I'm an instructor at Dr. Vikram Khurana's laboratory at Brigham Women's Hospital Neurology. And today I'd like to talk to you a little bit about gene therapies for cerebrospinal ataxias and other disorders. I'm very grateful and for your flexibility for me to be able to record this video a few days ahead. I'm actually going to step into the plane in a few hours to fly to Poland to visit my father and my 101-year-old grandfather. I haven't seen him in 3.5 years because of COVID, and I'm very grateful for the scientific discoveries that allowed me and my 4-year-old daughter to be fully vaccinated now so that we can actually go together and visit my grandfather in a very safe way. Today, I'd like to talk to you about gene therapies for neurodegenerative diseases and cerebrospinal ataxias. So I'm going to share my screen. And I've been working on gene therapies throughout my career, and it is actually a great pleasure to be here now at Neurology at Brigham Women's Hospital, because I feel the work that's been done and ongoing here is amazing and has such potential for transformative medicine and benefits of the patients. So it's a great place for me to be here and also share my vision with Dr. Vikram Khurana, where we're really trying to do research to benefit patients. So what is genetic disease and what is gene therapy? Genetic disease is actually very much implicated here through this Silva family tree. Basically, tremendous work to be done to actually put this together really shows that what genetic disease is, which is a very specific disease that's being passed on through generations due to a specific mutation in one gene. And so, and what is then gene therapy? Gene therapy is actually a strategy that hopefully can be actually beneficial to patients who carry such genetic diseases, because what it does is can target specifically that gene and maybe repair it. So there's actually a clinical trial ongoing that started earlier this year, which we're very excited about for SCA3. And this is a gene therapy clinical trial. I'll talk to you a little bit more about it later. But here, this is an implication of how this trial, how this kind of works. It's basically that we will have a gene therapy design here through antisense oligonucleotides, so synthetic pieces of DNA that can bind to a gene, the SCA3 gene, the gene that carries the CAG expansion. And by binding, it actually, through its mechanism, what it can do is actually can degrade this gene. So this gene is not functional anymore. So then the CAG expansion that causes the disease is not present anymore in the body. So that's gene therapy. The idea is that it's targeted and it's specific to that gene to fix that gene in a way. There's been actually tremendous progress happening in a few years regarding gene therapies for incurable brain diseases. And in early years, we really focused on actually developing the tools and making good and safe tools. There have been some clinical trials, which some of them have not been successful, which then actually kind of helped us to revert and go back and improve our tools to really make sure that what we're doing is really benefiting the patients by targeting the right genes and being specific and safe about it. And then in the recent years, gene therapy in the form of antisense oligonucleotide, that's predicted here in this graph, has been very fruitful. And clinical trials have actually resulted in FDA approvals for multiple diseases and multiple treatments available now for either Duchenne muscular dystrophy or SMA or even specific subtypes of Button's disease. What's very excited to us, made us very excited, is that earlier this year, there was a clinical trial started for gene therapy for SCA3, and I'll tell you a little bit more about that later. So what is an ASO and how does it work? ASO is otherwise known as antisense oligonucleotide, is a synthetic DNA, piece of DNA. And what it does, it alters our gene expression. So every cell, as you know, has DNA, and then genes are being kind of made from that DNA. And so what ASOs can do, they can do two things. One of them is they can actually shut down our genes. Because they're specific, they can shut down the gene that we want them to shut down. And so they do that by binding to the DNA and degrading that piece of DNA that's responsible for that gene. And so there's actually a few clinical trials ongoing with this kind of strategy of shutdown, therefore lowering the levels of this gene, this protein that causes the disease. And those are for Huntington disease, SCA3, and ALS. A second strategy, it's actually a strategy that causes a gene is still being made, but that the mutation that's in the gene is being skipped. And what happens is that ASO here binds to this mutation, to this area where the mutation is, and that area can be then excised from the gene, and then the gene is still being made, but it's missing that piece of the mutation, therefore the mutation is not there anymore, so the gene is again normal, or as close to normal as possible. And so actually this strategy has been very successful for many diseases which are already on the market now for patients to use. And this is for SMA, for Duchenne muscular dystrophy, or Batten's disease. So why are ASOs so promising? They're very specific, so we can really target, but also they're actually very easy to manufacture. And the chemical moieties which are around that piece of the synthetic DNA actually have been recently tremendously improved to induce a very good absorption of this ASO into the cells, distribution throughout the body, and stability, and most important, safety as well. So that's why this field has been actually booming in recent years. So how is this gene therapy applied? It's actually very simple. For such kind of complex system, the application is actually very simple. It's applied through intrathecal injection into the lower back, in this kind of between spinal cord space. So we basically do an injection here of these compounds, the ASO compounds, and by using the body's own way, how the CSF, cerebral spinal fluid, goes between the spinal cord and the brain and goes into all the places, we actually now use the body's own way to deliver this drug throughout. So then basically this drug goes all the way from the spinal cord to the brain. And using the system, what we can then ensure is that this gene therapy, which needs to target all the cells in our central nervous system, can actually go into the space and do its job and bind to this mutated gene, in this case, for example, the SCA3. And now I'd like to give you an update of our progress on developing these SCA3 isogenic gene-corrected control cells. So as you know, Dr. Vikram Khurana was able to collect fibroblasts from your family, from the members of your family, from Mark and Veronica, and with their cells having either 62 or 65 CEG expansions. And we were able to generate iPS cells, and now we have them growing in our lab. And we sent these iPS cells to Harvard Stem Cell Institute core to be gene corrected. So as you can see here in the graphic, we are with this new technology that Harvard Stem Cell Core collaborated with us thoroughly, and we really got this now working pretty well, I think, in their hands, to actually decrease the number of CG repeats from 62, 65 to 10 CG repeats. So this way, the cell line should actually have a number of CG repeats that a normal healthy line does, with everything else being exactly the same in the cell, but this gene mutation is actually now being fixed, or gene corrected, we call it. And so the Harvard Stem Cell was able to generate these lines, and we have basic QC already on them being positive. And now we were able to send these lines to Garvan Institute in Australia for whole genome sequencing to confirm that these lines do indeed have this healthy amount of CAG expansions and the SCA3 gene, and also to make sure that the lines are clean. And so therefore there's no gene edit, unwanted gene edit effects. So this is, we should be hearing soon from Garvan, and we're very excited to work with these lines to, one, really pinpoint the cellular pathology that is dependent and caused by the CG expansions, and two, to be able to test the ASOs that we had received from University of Michigan that Sharon was working on already, to be able to show that these ASOs indeed can bring actually the pathology and reverse the pathology back to the normal levels, that a line carrying the CG expansion of a healthy line derived from a patient originally is actually showing, so that this phenotype is truly a true rescue. With this, I'd like to thank everyone for their contributions to this work. It's been tremendous collaborative work from many different labs on the project with the DRPLA, and the funding on that through Cure DRPLA, and also now funding for us generating the iPS-derived lines from Silva Ataxia Foundation. I would also very much like to thank the Silva Ataxia Foundation and Silva family for supporting our work in our lab and at the Movement Disorder Division, and at the Brigham and Women's Hospital to really push this research forward for us to be able to expand the horizons and to help patients. Thank you again for your for acceptance of this unusual format of this talk, and I really wish you a wonderful meeting. Bye.